Okay, science for G-Man, take 3, and this is going to be about DNA. DNA, or to give it its full name, it's deoxyribonucleic acid, which tells you it's an acid. It also tells you it's got a deoxygenated ribose sugar in it as part of its structure. What else do we know? Well, we know that it's found in all cells. Let's draw a couple of cells. Yep, it's found in bacteria, our prokaryotes. There's a little bundle of DNA. Yep, prokaryote. It's also found in our eukaryotes, inside a nucleus which has a double membrane. Yep, these are our eukaryotes. Their lovely nucleus that contains all of the DNA. Part of the explanation for this in evolutionary terms is that these used to be 2 separate bacteria, and this bacteria invaded another one and took control of it. Hence we have our original cell's membrane and the invading cell's membrane enclosed by yet another membrane, the double membrane. So anyway, that's one small piece of evolutionary evidence. So that's, what else do we know? Right, well, let's take humans as an example. DNA example. What can we say about it? Well, it's a very long, very strong chain, and if we laid it out flat, it's about 6 meters. That's 20 feet in length, and that length is broken down into sections which are called chromosomes. That looks like a capital C, it's not meant to be. Chromosomes. And in humans we have 23 pairs. One from each parent, which is the only reason this number matters, because if you're going to have 2 parents, they need the same number of chromosomes so that they can match up and form those pairs. A different number wouldn't really work. However, the thing that makes the difference to the species is the actual DNA. So let's have a little look at the structure. I had to do a little bit more preparation for this one. And here we are. Here's our basic DNA structure. There's another bit. So DNA, we refer to it often as a double helix, which just means it's a ladder-like strand, like this, like a ladder. It's being coiled, twisted around itself. That's what the helix part means. And the 2 strands, look, are joined together by 4 nitrogenous bases. Let's worry about what they are. So what have we got here? This is our structure. So we have a G and a C. Well, C stands for cytosine. G stands for guanine. Or at the other end we have the T for thymine. And the A for adenine. Now these only ever form pairs in this order, which is why I always like to use the way of remembering it as a GCAT, because that tells you GC joins together and AT joins together, and it also reminds you that we have 4 base pairs. Yeah, 4 base pairs. So this is just one bit of a chemical. Each of our 4 base pairs, our nitrogenous bases, are, incidentally, they're all Where can I put it? Oh, there we go. This will do. Nitrogenous bases are hydrophobic. That means they repel water, and that's very important to remember because all of our DNA is in our cells, dissolved in the water in our cells. So which means that the central part of our DNA, our long polymer, our very long 6-meter molecules of DNA, is repelling the water. So it gets trapped inside this double helix. So the water can't get to the inside of it. So it can't affect it. However, we do know that our DNA goes into solution. And if it goes into solution and all the parts floated apart, we would no longer have this long chain, this long molecule that keeps the order of these nitrogenous bases. Now, the order is maintained by this bit along the outside, which is the sugar phosphate backbone, so called because this here is a sugar and this here is a phosphate. And the phosphate's quite interesting because if we actually looked at it, what you'd have sticking out the side like that is an oxygen. Oops, it's doubly bonded. Don't worry about that. You'd have another oxygen with a little minus sign. And that's because when it goes into solution, what happens is this was previously bonded to a hydrogen. But when it dissolves in the water, the hydrogen floats off, becomes positively charged, forms like a relationship with the water. And it's this that makes DNA acidic. That's why we call it an acid, because an acid is just the number of these hydrogen ions that are in our solution, in our water. Okay, so we've got this sugar phosphate backbone made up of sugars and phosphates. Each sugar phosphate is attached to one of our nitrogenous bases, which gives us, let's go for this section here. So that there, nitrogenous base, sugar and a phosphate. This is what we call a nucleotide. Oops. And it's the sequence of nucleotides that carries the information in our DNA. Yeah, this is a monomer. And DNA is a polymer. That means it's made up of lots of monomers. So we have 2 single strands carrying our DNA's information. Now the thing that holds them together is between each of the base pairs. These are base pairs because they form the pairs. Remember, GCAT. Yeah, and they are bonded together. Oh, I've just scribbled over it, with hydrogen bonds. These are quite strong, but they could be broken if the water could get in. But they're hydrophobic, so the water is repelled out. The water, on the other hand, is quite fond. It's attracted to, remember, the outside bit, these phosphates. So the water keeps this in a solution. However, the water doesn't get to the middle, which is hydrophobic. So the sequence of our nucleotides is maintained. Now, let's have a little bit more of a think about this. What else do we need to know about our DNA? Ah, yes, we need to know that each, for 3 of these here, so 3 nucleotides are called a codon. And they're so called because the 3 nucleotides match up to a amino acid. So each codon is around 20, matches up with an amino acid. Now, you've probably heard of amino acids if you've done any kind of bodybuilding and things like that. Amino acids are what we need if we're going to make proteins. So the way it works is each 3 codons form a sequence, and for every sequence of codons, so each sequence of codons makes a protein. Can you guess what it's going to be called? Should be an art. Protein is called a gene. Yay! So that's where the genes are. So we have a sequence of these codons which makes a gene. Now, what other information can we say? We have our long polymer, which is broken down into sections called chromosomes, and each chromosome is made up of codons, and every sequence of codon that codes for a protein is called a gene. Right, we've worked out that it's called a double helix because the core is hydrophobic, it repels the water, and when you put our DNA into water and it goes into solution, the outside of it is attracted to the water. It has a bit of hydrogen that floats off, and the water can't get to the core to break it apart or anything like that. It maintains our sequence of codons, our genes are all preserved, and lots of other stuff I think we'll have to come to in a later lesson. But one of the pieces of evidence, I must say, this in itself, so DNA is evidence for evolution, because all living things use the same system. Use the same molecules. Okay, and yep. Oh, I know what I can do. Let's have a quick think. What time are we on? Oops. 13 minutes, right. So I've said your DNA is in your cells, and you're all happy with that. You've got some nice pictures. So let's try and see if we can work out an experiment for you. So what you're going to need, you're going to do your own experiment. We're going to extract some DNA. You're going to need strawberries. So probably can't spell. Strawberries, salt, methylated spirits. What else we're going to need? Oh, you're going to need some water. Sandwich bag. It's a little plastic sandwich bag you can seal up. A sieve. Can't spell. Someone's bound to tell me that's wrong. Sieve. And yes, of course, washing up liquid. Okay, so what are we going to do? What we're going to do is I'm going to grab another sheet of paper, we've got our experiment. This is our apparatus. What we're going to do is we're going to get some strawberries. So we're going to take the strawberries, we're going to slice the strawberries. Yep. 2, we're going to make a solution. So, oh, let's add a bit onto our apparatus. Yeah. Let's have glasses. They look like that, you know, glasses made of glass. Make sure it's clear. So you're going to make a solution of water, salt, and wash liquid. And the reason you need that is because if you think of our cells, which are all bound together with the membrane made of phospholipids, you've got stuff that doesn't like mixing with water. So we need some wash liquid to make those bits of the cell break apart and be able to mix with the water. That's going to release our DNA. The salt actually lowers how soluble the water is. It makes it less soluble, so it's easier to get hold of our DNA, which is in solution. And the water is just a solvent, just to mix these together. So that's nice and easy. So we get the sliced strawberries. 3. We're going to put some sliced strawberry in our sandwich bag. And add some of our solution. Not much. You're going to need to be able to keep this all inside the bag. 4. Mash the contents of the bag with your fingers. It wouldn't hurt if you'd mash them up quite a lot before you even put that in. So you're going to mash that content up very carefully, like this. You know, mash until eventually you're happy that you've mixed it all together. So essentially we broke all the cells apart. 5. We're going to get our sieve, and we're going to sieve the contents of the bag. Whoops. Into our glass. So you're going to have a nice pinkish, possibly a little bit frothy, nice pinkish liquid. Now, also a part of this step, let's add another thing on. Let's just get a spoon. Okay. Mash some of the strawberry through the sieve, and then we're going to have the magic happen. Number 6. We're going to add some maths to our glass, and that should look like, in cross section, we've got our jus here, that is our solution of DNA and stuff. We're going to pour in our maths. Let's just do this in a different colour so we can see it, because maths, at least it is in this country, it's a different colour. We just need to pour it in so that it sits. And it's going to float on top of our solution like that. Glass and wait. And what you should see happen is, see if I can use a different colour. No particular reason why I'm using any of these colours. What you should see happen is a layer will float on top here. Yep. Now if you get the handle of your spoon and you dip it whoop, the spoon to extract the DNA. This will be the DNA floating on top of our methylated spirits. Now, if you ever read this instructions online, or you find them, generally speaking, you'll have people saying use very pure alcohol, like rubbing alcohol, things like that. But I found that meths works much better, which I believe is because it's actually a smaller molecule, so it has a better effect on the overall solution. And with it being coloured, it allows you to have a nice line which lets you see the DNA floating on top. And when you pull the DNA out, it's going to look a little bit like some snot, and hopefully that's not ruined the experiment for you. But anyway, feel free to get on with it.